all of you for coming out today. I appreciate it. It's probably the only time I'll give my talk to someone that doesn't have a shirt on and like a dog in the room. So I wish that all my audiences were like that. So my name is George. I'm a master's student at Cal State Northridge, and as Katie mentioned, I'm one of the Wrigley fellows out here. So it's a really great program. Even though I'm not a USC graduate student, I'm able to come out here and conduct my graduate research. So my thesis work is looking at the effects of predation risk or fear from predators on prey reproduction in a temperate reef fish. Some of you may recognize this little fish down here. This is a Bluebanded Goby, otherwise known as the Catalina Goby. They're very abundant. There's tons of them out at Catalina Island, so it's really the perfect place to conduct my particular study. Before I get into all the nitty-gritty details about my research, I'll just kind of tell you about my background and the processes that I took to get to this point today. So, sorry about that. Okay, I'll just go over here. Okay, here we go. So, first of all, I'm not from California. I'm from Maine, which is about as far across the country as you can get from this point in Los Angeles. But I grew up along the coast, and I loved going and looking at tide pools. So we have tide pools out here in California. This is a pretty iconic picture of a tide pool back in the northeast U.S. So I love being able to go out and play with the critters in these kind of shallow pools along the ocean. I also love fishing. Kind of gave it away here, but this is me as a young lad. I caught this was in the local newspaper, the York County Coast Star. I caught a 42-inch striped bass. I was very proud of it at the time. But that was back when I was about this tall, so it's really not that big. If I held it up today, it would look a lot smaller. But anyway, love tide pools, love fishing. So at a very young age, I developed this affinity for the ocean and ocean systems. So I went to get my undergrad degree at Northeastern University in Boston, Massachusetts. And they have a really unique program at Northeastern called the co-op program. And so through that, I was able to do some research experience as an undergraduate, where I took entire semesters off from school to go work in laboratories. So I knew I was kind of interested in science. So my first co-op experience, I was at the Cambridge Water Department. So I was going around and collecting water samples and testing them for different types of bacteria and stuff like that around the city. And then after that, I went and worked in Woods Hole in Massachusetts at the Marine Biological Laboratory. And I was working with this organism right here. This is called a rotifer. So it's a type of zooplankton. We all know, or maybe some of us know that plankton are highly abundant in the ocean as well. And some plankton are able to produce their own energy using the sun's energy, and those are phytoplankton. These are zooplankton, meaning they eat other plankton. But we were doing some really cool research looking at human aging pathways by using these rotifers as a model organism, similarly to how we use mice to study humans as well as model organisms. That's what I was doing with these rotifers. And also when I was at Northeastern, I took advantage of the thesis program, and that was a year-long marine biology course load that allowed me to travel around the U.S. and also internationally. And that's really where I literally got my feet wet doing kind of marine biology research. This is me doing some intertidal sampling in Massachusetts, and then this is me at the University of Washington's Marine Research Station up in the San Juan Islands in Washington State. That's really where I got interested in scientific diving. So now I just have a little map here of places that I've worked and done marine biology research, and I've been extremely fortunate. I call them working vacations. My dad calls them just vacations, but I have been able to travel around the U.S. and meet a lot of interesting people related to this field. So I started out in Nahant, Massachusetts. That's where I started the 3Cs program. I then went to Bocas del Toro, Panama, and Friday Harbor, Washington. And my very first job when I graduated from college, having never been diving in California, I somehow landed a diving position where I lived in Carlsbad, and I dove out of Orange County, out of Dana Point. And so that was kind of my first introduction to professional scientific diving. That was back in 2014 when it was really cool to have long hair. That's me here. That's just a type of algae. Anyway, so after that, I went to Dauphin Island, Alabama, So I was doing an internship in a Fish Ecology Lab. And so I was studying the interactions of fishes with each other, with their environment, and also kind of looking at the human impacts on fishing and fisheries. So any sort of fishing for profit is a fishery. And so I was able to do a lot of different types of sampling and collect lots of fish, some small fish, some big fish, and even some sharks. Pro tip, if you catch a small fish and you want to show it off and make it look bigger, hold it really far away from your body. So these fish, they're pretty big, but they're not that big. Not as big as they appear there. Anyway. And then after that, kind of revisiting the job that I had when I was an undergraduate, I was able to go back and work at that job back in Woods Hole with those rotifers again. So it was really cool to kind of reconnect with my former supervisor, and it was a perfect segue between Alabama, where I was applying to go to graduate school, kind of that gap timing. And so all this is to say that from each of these work experiences, I kind of figured out what I wanted to do professionally. So from the first job I had, I really fell in love with scientific diving. The next job in Alabama, I was really interested in fish ecology. And then lastly, this whole idea of using model organisms to, like rotifers, to study maybe larger, more complex organisms was really interesting to me. And so I've been fortunate to combine all three of those aspects into what I'm currently doing now as a graduate student. So now I'm in graduate school I'm going to my third year at Cal State Northridge. We call it CSUN. And I work in a fish ecology, Matadors forever. Exactly, very cool. So I'm in a fish ecology lab again, and it's a pretty wacky group. So this is our field crew 2 summers ago. This is my third summer out on Catalina. I really love it out here. And it's kind of like a work hard, play hard environment. So this is us. We're actually in the room directly above where we are right now in one of the laboratories working, a big group here. Can we do a little I Spy? Can anyone tell me what's wrong with this photo? Being a laboratory setting? Yes. What do you think? Maybe just stretch it. Facing the wrong way, exactly. It's terrible for photographers. Exactly. Anything else?
No goggles.
No gloves? That's
No goggles.
Oh, no goggles? Yeah, no goggles, no gloves. Do we have one over here?
You guys need flip-flops.
Need flip-flops. Don't tell Kelly that. Lab manager. I was thinking more, those are all great, and I didn't really think about those, but there's a little water bottle over here. You're not supposed to have water bottles. But I put this photo up as an example of what not to do. So I purposefully included the water bottle in the photo. But we're all working very hard. But we managed to have some fun too. We kind of let loose. If you have the right crew, work doesn't really seem like it. And this is just another photo of us all on the dock in a bunch of cages. Okay, so this is what a typical workday looks like for me. I spend a lot of time out in the cove right here, kind of surveying these reefs that I've set up for my thesis experiment. So now we can get into that. So as I said before, my research is really focused on looking at the effects of predation risk, or kind of the fear that predators can elicit towards prey on prey reproduction in a field setting. And so I work with this species, the Bluebanded Goby, but it faces risk of being eaten by a whole suite of predators out in the marine environment. It's a super tiny fish, kind of at the bottom of the food chain, so it is, scared constantly by larger fish out in the open ocean. And so before I can delve into the whole marine aspect of this, I want to take a step back and just talk about these kind of fear effects in general. So when I first was developing this project, I realized that there wasn't a whole lot of information about the effects of risk on marine fishes. So I had to go towards the terrestrial systems. So predators, they can influence prey fitness and prey reproduction in 2 different ways. There's lethal predation. This is a pretty iconic example. This is a lynx that has caught and killed a snowshoe hare. And so if you imagine it has effectively removed that hare from the population. And so it can no longer reproduce, and that might have some effects on the subsequent populations and generations down the line. But there's also this idea of sublethal predation, where predator characteristics or fear alone can alter the spawning behavior of prey. You imagine in that example I just showed, all the hares that witnessed the capturing of that one that was removed, that weren't eaten themselves, there's probably some learned type of behavior where they say, hey, I want to kind of avoid those situations where those links are present. It's actually, remarkably, it's been studied a lot in birds. And so that's kind of where I started this whole search. It's been shown that in situations where predation risk is high, birds will they'll lay fewer eggs, and the eggs that they lay will usually have higher death rates, higher mortality. So there seems to be some sort of effect on reproduction. So this has been looked at a little bit in fishes. This is a black goby, and the way this goby reproduces is females will lay nests of eggs, and males, shown here, will guard those nests of eggs, and they typically lay them in abandoned shells. So this is a mussel shell. You'll probably see them in the touch tanks later, but every single one of these white dots up here, that is a black goby egg that's being guarded. Okay, and so it's been shown in black gobies that when predation risk is high, they lay fewer nests of eggs. So you can imagine that has some effect, again, for populations down the line. But in general, a lot of the work that has been done with marine fishes has been done in laboratory settings. So maybe not as applicable to what we might expect to see in a natural environment. And that's mainly because it's hard to isolate risk cues in the wild. It's really hard to isolate those and concentrate them in one area because predators tend to move around, right? We know that fish move around. So that's really where my study comes in. So I'm trying to fill that gap, that knowledge gap we have, and I'm wondering, do prey alter their reproductive output in response to predation risk, specifically in the field? So it's been a big undertaking, and there's lots of things that can go wrong with a field experiment, but it can be very informative. And so what I've been able to do, I've been able to kind of study this question by using cages. I've been able to manipulate predation risk out in the cove right here with different types of cages. So as I mentioned, I work with the Bluebanded Goby, Lythrypus dalli, and they are a great model organism for studying reef fish population dynamics, mainly because they have a short lifespan, and it doesn't take them much time to reach sexual maturity. So they reproduce multiple times per year, which is great for me because I'm only available to come out and do my research during 3 months in the summer. And so I can run replicated experiments during a single field season, which is really nice. And this, if you have seen blue-banded gobies before, this is typically how you will find them. They are oftentimes perched on top of reef structure, and they're exposed here. And they can either be fully exposed, as seen here in some of the background gobies, or partially exposed here. And this is really important for blue-banded gobies because they're highly social. Given that they have such a short lifespan, they really need to take advantage of the time that they are around in order to kind of spark these reproductive events. And so you have individuals that need to be interacting with each other, and they can't do that if they're hiding inside of reef structure. So a little bit about their reproductive strategy. They are oviparous brood spawners. That's a fancy term that just says that the females will lay nests of eggs, males will fertilize those nests, and then guard them. This is a blue-banded goby, a male that is guarding a nest of eggs in an abandoned snail shell, and every little black dot you can see there is an individual goby egg. Also been shown that they'll spawn in artificial nests. So I don't know who found this out. I'm very thankful for them and their work, but they pretty much found that if you take a PVC tube and you line it with an acetate sheet, and acetate, someone explained this to me, it's kind of like the paper that you would put on something called an overhead projector. I don't know what an overhead projector is. Yeah. There's been, yeah. So if you take one of those acetate sheets, you cut it up into a rectangular piece, you can line these PVC tubes, and the gobies will actually go into those nests, and they will lay eggs. And so because it's lined with a piece of paper, we can actually go and we can sample those nests without being so invasive or destructive. But this is a male blue-banded goby doing the same thing that it would if it were in an abandoned snail shell or some other sort of natural nest. It's guarding those nests of eggs. And this is what those nests look like. So when we pull out that piece of paper, we unravel it, you take a photograph of it, and every little individual red dot is a blue-banded goby egg. So this is really the key to my study. It allows me to actually measure the total number of eggs that are laid, so I can get a sense of reproductive output directly. So their primary predator is the kelp bass. It is the most abundant piscivore, meaning fish eater, at the site where I'm conducting my study, right in the cove here. And it's also been shown that blue-banded gobies respond to predation risk by changing their behavior. This is kind of a confusing graph, but I just want you to pay attention to these two rectangular boxes here. So my advisor, Mark Steele, he did a lot of work on blue-banded gobies when he was getting his PhD. For the Star Wars fans out there, we call him Goby-Wan Kenobi. He's kind of like the Goby stage. And so he showed that when predators are present, blue-banded gobies spend a greater proportion of time hiding under rocks or hiding within reef structure. And when predators are absent, they spend more time perching and exposed, which seems kind of intuitive. When predators are around, you hide. When they're not around, you don't hide. But we have to do studies like this so we can kind of build a foundation for future studies like my own. So I'm thankful to Goby-Wan. So as I said, I'm doing a caging study. And what I'm doing is I'm using different types of cages to manipulate the physical separation between our prey species, the blue-banded goby, and predators. This is a kelp bass. It's just a generic that could represent any predator for our purposes here. So what I did was I first had to go out and make these rocky reefs. That's kind of my little rock rubble pile down here. And then I put 5 of those artificial nests that I showed you, the PVC tubes, on each of the reefs. And then from there, the thing that I changed was the caging type. So in here, this is my low-risk scenario. And so this is from the perspective of the goby. The predators, the shading here represents netting to exclude predators. And so in this situation, I was thinking, gobies might perceive this as a safe environment, a low-risk environment. So from there, I had a medium risk treatment where it was like smaller cage dimensions, but still excluding predators. Predators can't move on and off the reefs. And then lastly, have a high risk environment where the side panels of the cages have been removed, so predators can move on and off the reefs freely. So to each of these reefs, I added 20 gobies, 5 males and 15 females. 5 of the females were pregnant. Because I'm running a reproductive study, I wanted to make sure that I had similar starting populations on each of these treatments. And these, the ratios of 5 males to 15 females is representative of what we usually see when we go out and we collect gobies. Hi, welcome. Let these people sit down and I'm going to grab a drink. It's all good. The best is yet to come. Oh, good. Okay. So, these are my 3 reef treatments. My 3 different risk treatments. Now I can show you kind of where I'm conducting my study. So some of you may recognize this area. I probably rode in on it today. So this is Big Fisherman Cove. It's a marine protected area. And I collect all my gobies from Isthmus Reef. Don't worry, I left a few of them out there. There's a few blue-banded gobies still at Isthmus, but I did collect a fair amount this past summer. And this is a really nice place to conduct a study like this because being a marine protected area, there's no fishing, there's no anchoring, so I can go out and dive here and not really have to worry about people buzzing me or dropping anchor and stuff like that on. And so I had 18 reefs in total, and I constructed them along the southern edge of the cove here. And thankfully, nobody anchored on any of my reefs this summer. It was awesome. So that's happened in the past, but this summer we were. And so I had 6 of each of my 3 treatments. And so I did 2 types of surveys once I got my experiments up and running. So I had Ness Surveys, where I would go out after the 4th day that I deployed gobies onto the reefs, and I would check those artificial nests for eggs. So this is a picture of me pulling out those acetate sheets. This is what those sheets look like underwater when you actually find eggs on them. And we would photograph those nests when eggs were present. So this is my dive buddy Hunter holding a nest on a slate underwater and me photographing them. And so again, this is what those nests look like. And what I do with these photos, I take them back to my computer and I count every individual egg. I'm trying to find ways to streamline that process. If anyone knows how to use Python, it'd be helpful. After this. And I also did density surveys where I would swim along my reefs and I would count how many gobies that I could see exposed on the structure. So that was kind of to get at a little bit of the behavioral differences between low, medium, and high risk treatments. And I also, at the end of my experiment, I recollected all the gobies that were still on the reefs at the end of the trial. So I'll show you some results now. The results that I'll show you are from my previous field season. I haven't really started counting any of the eggs from this summer. They're a bit backlogged, but I do have some preliminary results that kind of I found qualitatively. So in general, it seems that there's lower reproduction on those high-risk reefs. This is a, again, complicated graph, but essentially showing you here the 3 different treatments that I have and the average number of eggs produced on each of the reefs from each of those treatments. So you see here is that in the low and medium risk treatments on the left, where predators were completely excluded by the netting material, there's really no difference in reproduction. But only on the high-risk reefs where you had full predator access, you saw a 33% reduction in egg laying. And so that's really important when you consider that these, on average, these gobies in the low and medium treatments are laying, 2,500 eggs per reef, and then you have something like 1,800 eggs in the high-risk treatment. So that can have a big effect, again, on those future populations. And so, sorry, this is interesting in itself, but it doesn't really tell us why that's happening. You might be thinking to yourself, okay, George, those high-risk reefs, they had open sides. Like, it makes sense. The predators were going on there, picking off the gobies. No gobies, no eggs, right? So next, we have to incorporate those visual surveys that I did, and I found that there were also fewer gobies observed on those high-risk reefs too. So either they were gone or they were spending more time hiding in the reef structure. Yeah, so overall, everything decreased over time in terms of the proportion of the population that I saw on the reefs, but the high-risk reefs, this orange line, seemed to drop off much more quickly. However, when I recollected all my gobies at the end of the experiment, I found equal proportions of gobies on each treatment. So regardless of predator access, it seemed like you still collected the same number of gobies. So that's pretty important, because it kind of shows us that it's more of the behavioral response and the fact that these gobies are hiding. I wasn't able to see them on my visual surveys. That was leading to these reductions in reproductive output, not necessarily the actual lethal removals. So revisiting my original question, do prey alter their reproductive output in response to predation in the field? My results suggest that yes, they do, but only in those high-risk treatments. So you need to have you need to allow for full predator access here in order to really elicit that risk response and to get a treatment effect there, where you had fewer eggs laid coupled with increased proportion of time spent hiding. And so revisiting that original picture I had in my photo, so this is what I would typically see in those high-risk reefs, where you had predators that were either resting or kind of waiting opportunistically for a moment where those blue-banded gobies could be kind of snatched up. I will say, spoiler alert, blue-banded gobies are very good at hiding and avoiding predators. So I don't think they were really I never witnessed natural predation. I never saw kelp bass or anything just go up and slurp up a goby. So for this summer's experiments, I kind of switched things up a little bit. I was trying to build on last summer's experiments. And so I extended my trial duration from 2 to 4 weeks, and that was really to get at the short versus long-term effects of predators and predation risk on reproduction. And so it seems that these effects and these reductions in reproductive output are persisting over time, which is really important. It's not like the gobies are habituating to predator presence. It seems more that they are just constantly afraid in those high-risk environments. And I also added an uncaged treatment, where I, exactly as it sounds like, I had no cage, no structure surrounding the rocky reefs. And it doesn't seem like there are any differences in between the high risk here and the uncaged treatment. I was kind of thinking that predators might either be attracted to or dissuaded by this high risk treatment here, but I really didn't know the answer to that, so I had to run that field. And it doesn't seem like there is any effect on the reproductive output of the gobies. It doesn't seem like there are any differences in kind of the grouping or aggregating behavior of the predators. That's kind of where I'm at right now. So what does this all mean? Why is this important? So it shows us that, for the first time, that these effects of predation risk on prey are substantial in nature in that these are things that, it kind of confirms what we already found in the lab, but there was no way for us to know that those types of factors existed in the field unless we went ahead and did it. And then also, it kind of speaks to the types of practices that affect predator abundance. So if we're realizing that predators are maybe having more of an impact on prey than just those lethal removals, those one-to-one interactions, we might start thinking about, okay, what are the types of things that we as humans are doing to affect predator abundance, and what effects might those have on the like lower trophic levels or lower steps on the food chain? Things like overfishing, where we're removing predators, or things like marine protected areas, where we are kind of supporting predator growth, things like that. So doing the study in the cove here, there's just a high abundance of large predators, right? And so I may have seen, you may see something different. I may have seen different results if I'd done this directly outside of the cove where predators are being removed. So with that, it's a field study. I have tons of people that have been helping me out. Lots of help from my lab. Also, all the volunteers that have come out to help me here with diving. One of the really cool things that I've been able to do through Wrigley and through this project is mentor a bunch of undergraduates. And so that has been by far one of the most fulfilling parts of my whole graduate career so far. And specifically, I've been able to work with undergraduates from 3 different universities, which is really cool. I think there was a moment this summer where it was a all-in-one dive boat diving together. It was myself from Cal State Northridge, another graduate student from Moss Landing. There was an undergraduate from USC and an undergraduate from Loyola Marymount. So you had this all people from different types of backgrounds, different levels of diving experience, all working together to conduct research, which was really cool. And specifically, I've been able to work with some of the USC REU students, so that's Research Experience for Undergraduates. It's a really great program that they have out here at the Wrigley Marine Science Center. And yeah, this student I was working with this summer, Hunter, actually just presented his findings at an undergraduate research symposium. So it's been really cool to kind of facilitate that training and that knowledge, and just be a mentor and kind of see where these undergraduates will end up next. It's really been great. And again, all through Wrigley, it's made that possible. I want to thank the facilities here. All the waterfront staff have been, they're pretty much scientists, although they don't have that title. They are really the ones that they're kind of pumping out these projects as well. And then all the funding organizations have been very fortunate that these people have been willing to support me financially. Whew. Okay, and with that, I thank you for your attention, and I'll answer any questions you have.
Look at the colors. How can you hide when you have that?
Right. So the question was, correct me if I'm wrong, the question was, they hide a lot, they want to avoid being eaten, so why are they colored like that? Why are they so fluorescent? It seems like they could easily be picked off by any sort of predator. It's a really great question. I'm not sure about the evolutionary benefits of being that color. I think the only thing that I can think of, and what makes sense to me, is that they need to be interacting with each other. Now, granted, they live kind of within close proximity of each other. But yeah, definitely not an advantage to be that color when you're trying to avoid being eaten. But I think it must be something for interactions with other gobies. Wow, that's the only thing that I can think of.
It's really sexy.
Yeah, that's why I work with it. Yes.
I'm just sort of curious about the impact on reproduction. Is it that, the fish have fewer opportunities to have these romantic interludes, whatever, with each other? Or is it more that, you could have something like, do fish experience stress, what we might experience with cortisol, things like that? Could it be some biomechanical?
Right. So, so kind of getting at, so that was the purpose of extending that trial duration was to see if it was some sort of physiological response. Other things that might factor into that, so if they're spending more time hiding, they can't be interacting with each other. They're probably eating less food. They're bringing in less energy. It's pretty energetically costly to produce that many eggs. So it could be, some sort of endocrine response, some sort of stress response that's going on there. I think it's probably a combination of the two. Something like I'm not interacting with other gobies, and also I'm really stressed out, so energetically costly to be worried all the time. Yeah. Yes.
The predators, their eyesight, would they see the colors?
Yeah. So I think, especially being in out in California in these kind of temperate systems where visibility is somewhat questionable at times, I think it's particularly important to have those like visual abilities to pick up on prey. I have seen a few like near misses. Like I said, I've never seen a goby eaten in nature, but I've seen predators kind of dart at gobies. Sometimes they miss. So I think it's probably a combination of visual cues and some sort of olfactory or scent cue that they're picking up on. But there has been a little bit of work done on this in tropical systems where the water is much more clear, and so I can imagine there might be some differences in kind of how the predators are conditioned to pick up on prey. Yes?
Did you wait for the predators to?
Yeah, so how much? Let me see. Do we have enough time?
We only have a
Okay, so to answer that question, yeah, so one of the interesting things about this design is that I couldn't really ensure that there were predators there at all times. I couldn't put a kelp bass in a cage. I was thinking about doing like a little cageception, a cage within a cage, but the predators aren't going to act normally in that situation. So I had some undergraduates do a little project. I've taken some time-lapse photos to kind of see how predators aggregate around the different treatments. I wanted to make sure that they weren't favoring one treatment over another. And so they found that they were being visited frequently by predators, so there was kind of a constant risk effect there, but there weren't any differences among the treatments. They were being visited by the same numbers of predators and the same numbers of each type of species. Actually, rock grass were the most abundant. Yes. Was there a question in the back too?
Oh, I'm not sure if I'm using the right terminology, but have there been studies done about like the level of consciousness of the gobies, like to actual loss of another goby or that sort of thing?
Yeah, I'm not sure. There's so they form these little social hierarchies. So I know they can pick up. There's been a fair amount of work done. I'd say most of the work that's been done on them that's not field-based. A lot of the laboratory stuff that's been done is on kind of the social structuring and which goby is top dog, which is the alpha goby, the beta goby. So they can perceive when individuals are lost within a population. Yes, but I'm not sure specifically for risk factors. I'd just be like, hey, the big goby left. I'm going to be the lead. What do you think?
One
more
question.
Okay.
What do the gobies eat? And also, did you do any between your years of different pollution levels in the water to see if that would have any effect on it?
Okay, so goby, the first question, they eat plankton. So they'll leave the rocks and they'll kind of bite at things and then settle right back down. So just things that are floating by. And there wasn't any limitation to food that I observed, even for gobies within cage treatments. And then I haven't really looked at pollution. I haven't added that as like a factor. There was a lot of other stuff that I kind of looked at, like temperature, position of the reef within the cove, stuff like that. But that would be interesting to consider. And I'm not sure what the levels would even be like in the cove.
Yeah, but look, it's pretty unpolluted here.
Yeah.
Pretty clean water, a lot of flow, but
Yeah, that's an interesting question. Okay,
Thank
awesome.
you so much.
Thank you.